I would bring you some of the stories. These are mostly stories that is related with the South Asian populations, how these populations who are present in this subcontinent, they got admixed, they have migrated from one place to another place. So how these genomic data are helping the scientists to discover many more new things, and also how some of the data sets at one moment when we generate the data set, when we interpret the data set, then we feel like, oh, this is the end of the story and we have done everything. That is something like we should get now the Nobel Prize. But after a few years, when we again work with a higher resolution, with a more number of population, then we feel we were wrong initially. So these kind of stories we would bring one by one. So it was 2011 when a leading newspaper, Hindustan Times, has made a survey in India, and they said that any two individuals living in India, they could understand their language. The chances of this understanding is 1.6 to 95%. What about genetics? Any two random individuals you pick from India, they would certainly share more than 30% up to 100% of their genomic ancestry. So DNA binds India more. So there is a saying in India that says that कोस पे पानी बदले 3 कोस पे पानी. The meaning of this, that every 1.5 kilometer the water changes, and every 4.5 kilometers the language changes. This is not because we brought several languages from different parts of the world. This is because the time we have lived in this subcontinent and the mutation in languages is much more higher than the genes. So the basic thing you might have already understood during the previous presentations, that the basic tools for a population geneticist are mainly three things. First is mitochondrial DNA, second is Y chromosome, and third are autosomes. So usually by using all the three kind of markers, we bring a population history. So for example, like if someone does just mitochondrial DNA, then because mitochondria is maternally inherited, so just by studying mitochondria, one can understand the maternal history going back to several thousand years. Similarly, for Y chromosome, one can understand the paternal history just going back several thousand years. These are more straightforward comparing to the autosomes, but as you can see that these individuals, they have not just one ancestor, they have multiple ancestors. So what about the other ancestors? So by using the autosomal, the biparental inheritance, one could understand the whole genomic history, but the autosomes is not as straightforward as mitochondria and Y chromosome. As you know, the recombination breaks, and every generation going back, we share 50% from our parents and 25% from our grandparents. So this is how this needs a lot of complicated mathematics. Otherwise, that one is more straightforward. So by using just 4 letters, A, G, C, T, the population is being sequenced, and these population are divided into several haplotypes. The group of haplotype is known as haplogroup, and these never applies, not always applies on human population, but usually these kind of trees where you can see with the time when we accumulate more mutation, the tree is longer. When you accumulate less mutation, trees are smaller, diversity is more. But these are not the hard facts, but most of the cases these things applies, but there are exceptions as well. So in 2005, when we started working on Andaman Islanders, that time sequencing the complete genome was quite expensive. So we have sequenced their mitochondrial DNA. And just by sequencing the mitochondrial DNA, we identified 2 independent lineages that was connecting them through the Africa. And by using these information, we concluded that there was out of Africa migration, something like 65,000 years back, and Andaman Islanders, they are the remnants of out of Africa migration. But again, in 2015, when we have used the complete sequences from multiple population worldwide, then we found that what we have expected, that just there was a single migration out of Africa, that is not at all true. There are multiple migrations out of Africa, and not only migrations, we have also admixed with the other hominin, that is Neanderthal and Denishowan, and there are many more unknown which we don't know yet, but with time we may know them because of the technological advancement. So the overall human migration theory, human origin theory is getting much more complex as we go deeper in their analysis. So this is one of the example of skin pigmentation. You can see here that when the skin pigmentation allele is distributed here, and where the allele is more, it means these people will be lighter, and these people will have a darker color of their skin. So during the evolution of the skin color, either one can see two things here. The first thing is either the skin pigmentation had originated somewhere in this vicinity and migrated from here to there, or from there to here. Our second thing is another model, that is isolation by distance. The isolation by distance model tells that usually a population moves in a certain area, but these population always cross another population territory. So this is how that a gene moves from one place to another place without the migration of individual. And whether this situation represent the migration of individual or the migration of or just the isolation by distance model, this is much more complicated than we have ever thought. So charting a migration needs much more information, and that comes from multiple things, not only genetics, that includes the linguistics, that includes the archaeology, that includes even many more things for the human migration. So the major point for interpreting the data is the data resolution. Like when we make the resolution of the data, that if some labs start to just screen a stretch of DNA that is something like 100 kb, and another lab scan something like 1000 kb, and then compare both of the data set by using the same parameters, then sometimes the result comes quite drastic, different from each other. So here I bring one of the story which we started initially. Something during the 2005-2006, overall, while working with the population genetics, when someone sees that, okay, the frequency is high in some population, then that means that probably there is nearly 80 to 90% chances that that particular lineage might have originated in that population. So this is the same case here, that there is a haplogroup. Haplogroup is kind of, one can say that a group of individuals, if they carry a stretch of DNA and that stretch of DNA has same type of mutation, then this is known as haplogroup. So when there are 4 population groups, so just to show the population group, let's come back to this slide. So overall in South Asia, there are mainly 4 language groups. So these 4 language groups, they are distributed in a very geographic manner. For example, like mostly the Indo-European groups, they are present in the western part, they are present in the northern part, even into the central part. So the Indo-European language family is covering a major part of the Indian subcontinent, whereas Dravidian, they are present into the southern part, with some of the Kuruk and others, they are present into the central part, whereas the Austroasiatic, they are present in the pockets, and they are into the central part as well as the eastern part. And there are the Tibeto-Burman, they are also known as the Trans-Himalayan population. They are present at the fringes of Himalaya and then the northeastern part. So you can see that there is no overlapping of the linguistic distribution. So can we also see this kind of distribution among the among the among the genes. So let's go back to this. So there is a haplogroup known as R7, and this haplogroup, if we compare all the language groups, then we can see that it has a very high significant frequency difference between all the languages groups except the Austroasiatic. This Austroasiatic group has a very high frequency of this language group. And also in this principal component analysis, that you can see that the R7 is making this haplogroup. R7 is making this population into a tight cluster, and because of that, this group is showing a very different cluster based on the haplogroup frequencies. So during 2005, 2006, these kind of analysis usually show that there are very highly likely chances that this haplogroup might have originated into the Austroasiatic groups and then later distributed to other language groups. So, yeah. Which one? The percent values in the table. Oh, this is the frequency. Frequency of that haplogroup in that language group. Yeah. And we basically focused on this, only R7. So whatever I talk, that will be related with the R7. So then that was based on a small stretch that was HVS1, hypervariable segment 1. So now we have completely sequenced their mitochondria. And here what you can see that this mitochondria is showing a very distinct pattern, that all the Austroasiatic is shown with this color. So all this Austroasiatic, they are showing a common maternal founder that has originated nearly 2,500 years back. Whereas this overall, this haplogroup is nearly more than 25,000 years older. And there are two distinct branches, R7A and R7B. And overall, if you look to the distribution of language group, then you can see that Dravidians as well as Indo-Europeans, they are present in both of the branches, whereas Austroasiatic are present in just one branch. It was most frequent in that particular language group, but it was present only in this supermother branch, which has given rise several smaller, very recent branches that has expanded quite recently. So overall, in the neighbor joining tree, if you look to the distribution of a large number of individuals, then you can see that there is one branch that is predominantly Austroasiatic, and another branch is predominantly Dravidian, and the Indo-Europeans, they are also present in this branch as well as in this branch. And overall, the distribution of this haplogroup in various language group, you can see that in Austroasiatic, this is just a much more recent comparing to Dravidian and Indo-European. Yeah. So there is a dating method based on the mutation. So we calculate the mutation rate of every stretch of DNA, and this stretch of DNA is being used for individual. We group the individual into the various language group, and based on that, we calculate the mutation. For example, like usually most of the mitochondrial mutation rate is nearly 7,421 years per mutation. And then you calculate the mutation, just mathematically divide that how many individuals you have. So this is just a very easy process. So in this case, one can see that all the Austroasiatics, they are sharing a very recent founder that has originated much more recently. This is something like Genghis Khan kind of expansion, where something has happened in the past that a group of people speaking Austroasiatic language, they started to mix with the population of this area. They married with the local women. So not local women, one can say that they married with only the branches that were present in the R7A1, and then they expanded very quickly. So this is a very recent expansion that was showing recent expansion followed by the founder effect, and that was showing a very high frequency of this haplogroup. So, and then further, yes. Yeah. More? Yeah. Clear? So we went a bit further. We used the detailed information coming from one of the linguistics. And here we have divided the whole Austroasiatics. So by dividing the Austroasiatics, this is the linguistic classification. This is not the genetic classification. So based on a linguistic classification, the Austroasiatics are divided into Northern Munda as well as Southern Munda population. And here you can see that most of the R7 are skewed for the one branch only. The second branch has very low frequency of this Austroasiatic linguistic group. So probably this group has, when this group has divided into two groups, this group has received the R7A, and there were some supermother. Because of the supermother, this group has expanded, and that is why the frequency has elevated in this particular population. So what is the most important thing here? That the number of samples plus the number of number of the stretch of the DNA. So if someone has studied just the HBS1, the small stretch of DNA, it was not possible to divide this into such a high resolution level. So by using the HBS1, one could clearly say that this haplogroup might have originated into the Austroasiatic, but by using the detailed information coming from the whole mitochondrial DNA, it was possible to differentiate it into various multiple haplogroups, into the subhaplogroups, and then by using these information, putting with the linguistic information, one can clearly say that this group has originated among the Dravidian population, and then a subset of this group has been received by the Austroasiatic people, and then just by the recent expansion, the frequency has elevated in this population. So this Austroasiatic is more intriguing. It is quite complicated population. Here, that is their maternal ancestry, that is their paternal ancestry. And this has also given something like a question of 150 years, because genetics came very recently, and only just within 5-10 years back, we could able to resolve this question. So what was the question? That this is the Southeast Asian population, and this is the South Asian population. This color represents the South Asian ancestry. This color represents the Southeast Asian ancestry. So just compare these two top figures, and you can see that the Austroasiatic population living into the Southeast Asia and Austroasiatic living into the South Asia, they are completely different in terms of their maternal ancestry. That is clear. The green color shows a South Asian ancestry. So Austroasiatic population living in India, living in South Asia, they have the South Asian ancestry. Clear? The Austroasiatic population living in Southeast Asia, they have the Southeast Asian ancestry. What about Y chromosome? So the Y chromosome has problems because the Austroasiatic population living in South Asia, they have nearly 30% Indian South Asian ancestry and nearly 70% ancestry which associate them with the Southeast Asia. This has created a lot of problems because when there was no detailed data, no Y chromosome sequence were present, so based on YSTR and based on some low resolution markers, people were trying to play with this. So a series of paper has been published just by trying to show that this color has not originated into Southeast Asia, but it has originated into South Asia. So this is another test that has compared the geographic longitude as well as the Southeast or East Asian frequency for the mitochondrial haplogroup, and this was for Y chromosomal haplogroup. And here you can see that there is no effect of geographical longitude for the Austroasiatic population in terms of their mitochondrial ancestry, and there is a clear-cut effect of their Y chromosome elevated frequency in terms of geographic longitude as well as the separation of Y chromosome and mitochondrial DNA. So because of that, a series of paper that has been published on that, and these papers, they all were trying to show, they all were trying to fit the Y chromosome in such a way so that the theory of mitochondrial DNA should be code for the Y chromosome as well. So anyways, but there was a big paper. It was not a paper, it was a commentary by Professor Aravinda Chakravarti. He's a very well-known professor in United States. And as something, if you bring some of the information which you can say that, let's say that if sun comes from east, you don't need the citation. So he has used the same thing, that the earliest occupation of the subcontinent was the Austroasiatic people coming nearly 60,000 years ago from Africa. So according to his writing, that these are the perfect facts, these are the, there is no debate on these facts, but in reality, that all these facts, what he has written is at the moment is completely wrong, because of the complete information comes from the whole genome. So my emphasis on these things, that when something comes from a smaller set of DNA, smaller set of individuals, then usually the chances for them are being wrong is very high. So just when you complete the detailed information which comes from the large stretch of DNA or the whole genome analysis, then those things are more robust. But still, one should look the other disciplines when it comes to the humans, because human origin is not as easy as for a something like a cow migration or something like any animal migration, because this is not very straightforward. We mix so much that looking for a smaller stretch of DNA, looking for a bigger stretch of DNA, these things makes quite complicated things. So these are two very most cited tools, and these are the basic tools which population geneticists use. That is the principal component analysis and admixture analysis. And by using these tools, many of things has been even interpreted very wrongly. For example, like, and also what is the question and what answers you try to use, what markers you try to use for answering those questions, that is also important. For example, in this case, the European gypsies, they are known as Roma. So everyone knows that they have migrated from India, because this is embedded in their folklore, this is embedded in their lifestyle. So there have been nearly more than 50 studies they have been done on Roma population. So here what they have done the genome-wide analysis, and by using multiple IBD sharings as well as other statistical analysis, they have showed that the Roma population are most closest to the northwestern Indian population. So here they have used nearly thousands of individuals, spent nearly $50 per sample into 1,000, and they could reach to the conclusion that said that they are associated with the northwestern Indian population. What we have done, we have taken their Y chromosome, just one haplogroup. We have used this 17 STR markers, something like 200 rupees per sample, 1,000 sample, and we could reach to the conclusion that these population are associated with the scheduled caste and scheduled tribe population of northwestern India. So the question and answer of that question is more important in terms of using the methodology. So what we have done, we have used the Y chromosomal details, and there is a very clear-cut signature of that Y chromosome. We were fortunate to find that this is a haplogroup H, and when we look to the haplogroup H distribution in India, we can clearly see that it has originated somewhere in the south and then expanded to the various parts of India. And here you can see that one group which was existing into the northwestern part of India. It has gone out of India nearly 1,400 years ago by molecular dating. So we use the YSTR markers. These YSTR were able to tell a recent ancestry. When we go back in the time, the YSTR fails. So, and we were fortunate to use the YSTR because we need to tell a story which was just 1,400 years older, not 14,000 years older. So we found that they were most closest to the population of Northwest India, but this Northwest Indian population were belonging to scheduled caste and scheduled tribe population. They were not at all closer to the other OBC and general population of Northwestern India. So, and then we went one step further by using more analysis, and we found that the North Indian population who are still nomads, they came from Rajasthan. They are the most closest population of the Roma population. So the same thing has happened with the Jewish population. There are two migration of Jews. One is Mumbai Jews, and again, second is Cochin Jews. Cochin Jews are the first Jew who came nearly 2,200 years back to India. There are some records related with that. Then Mumbai Jews nearly 1,100 years back to India. So most of the Jews who were living to India, they have gone back to Israel because Israel government they made an offer to the people of Jewish origin to bring them back to Israel. But still, there are nearly 5,000 to 6,000 Jewish people. They are living in India, and by using the detailed analysis here, you can see the principal by using principal component analysis. And here is the Indian cline. So Indian cline that contains the Dravidian population, Indo-European population. Here is Pakistani population, European, the Middle Eastern, the Caucasus, then Central Indian, the East Asian population. So this is a principal component analysis that is based on the allele frequency of nearly 500,000 of markers. So just by using this analysis, you can see that the Jewish population, they are just falling on the Indian cluster. And just by using only principal component analysis, one can't say that the Jewish population has any affinity with the population of Israel or with the population of Middle Eastern population. But just by when we use the admixture analysis, you can clearly see that the Mumbai Jews are here, and this ancestry, that is the dark blue color ancestry that is present in the Mumbai Jews in a high frequency, whereas some traces are also present among the Cochin Jewish population. So that has suggested that there is some link of Indian Jewish population with the Middle Eastern Jews, and this has the geospatial distribution analysis, because everything in population migration should be looked for two things. One is the spatial thing, another is temporal thing. So with time as well as the geography. So time and geography are two things. And here you can see that all the Indian population, they share nearly 0 to 9% of the ancestry that is associated with the Middle Eastern population, whereas there is an elevation of the ancestry is present among the Mumbai Jews as well as the Cochin Jews. That clearly says that this rejects the isolation by distance model for a population, for an allele distribution, and there is something which connects the Mumbai Jews and Cochin Jews with the Middle Eastern population.
In China?
Which one?
Green dots in China also?
Yes, because no green dots because this is 0%. So yeah. If there is some ancestry which is present somewhere, let's say there, that would decline, and you see something somewhere zero frequency of that same, because of the distribution of that allele into the time as well as in the space. Yeah. So that the molecular dating was nearly matching with the written records, which shows that this Jewish population has migrated to India nearly in two groups. One is 2,000 years ago, and another is 1,200 years ago. And here you can clearly see that maternal and paternal ancestry. So again, two different stories, because their maternal ancestry is much more closer to the Indian gene pool, whereas paternal ancestry, like these Mumbai Jews, they are more closer to the other Jewish population. So here, what one can infer that mainly the migration was male mediated. They had mixed with the local population. Then there was a lot of assimilation of the local population. That is why this frequency, this kind of ancestry came into the Jewish population, because of the admixture with the local population. But once they had a large number of individuals, they stopped admixing with the local populations. And here is another story related with the Siddhis. The Siddhis has been brought by Portuguese to India during the 15th to 19th century as a slave trade. And this is the African cluster. This is the Indian cluster. Siddhis are in between African and Indian cluster. They are more closer to Africa. That says that their ancestry should have been much higher for the African comparing to the Indian population. And here we compared their autosomal maternal ancestry, paternal ancestry, and one can clearly see that the autosomal ancestry shows that they are 60 to 65% ancestry is still associated with Africa. Nearly 35% or 30% of ancestry is associated with the Indian, including the European clime, as well as mitochondria is completely showing the opposite way. Nearly 60% is associated with South Asian, 30%, 40% is African. The Y chromosome is more closer with the autosomal kind of ancestry, where African ancestry is a bit higher comparing to the South Asian ancestry. So one has to understand the history to infer this. Otherwise, if someone gives me Siddhi sample and then I start analyzing this Siddhi sample, I can tell that there is some African population who came to India, but when it came to India, that is another question. So that is needed to be analyzed with the detailed information coming from the history. And our another story is associated with the Parsi population. The Parsi are the people who are the makers of modern India. There are few Parsis, they are living into the coastal region, as well as even Baghdadi Jews, as well as even the Baghdadi Parsis. They are also living into the Kolkata. We are still analyzing these samples, but we have analyzed the Mumbai Parsi samples as well as the Pakistani Parsi samples. And here the information again comes from multiple number of markers, multiple ways of marker. Even we have analyzed the ancient DNA for their maternal ancestry, and one can see that there is a clear-cut transition of Parsi, the genes of Indian ancestry, and the mixing of the Iranian ancestry. So the story of Parsi is quite interesting. That during the exile of Parsis, they are the Zoroastrian, and because of the Islamic conquest, they have to move out of the Iran, and a group of Parsi, they came to the Sanjan, that is the coastal area of India, and there was a local king. His name was Jadi Rana. So Jadi Rana sent a message to them by sending a glass full of milk. Then the Parsi has added the sugar in the glass, telling that the full of milk means my kingdom is full of the people, I can't accommodate you. Then Parsi just mixed the sugar in the whole milk. They said that we will live in mixing together with you people. So this is how the Parsi came to India. And there is no ethnic conflict with the Parsis. They are still living without any complication in Indians in India. So the study of Parsis initially, when we have done their principal component analysis, you can see that they are in between the Iranian population and also in between the Pakistani population. So just by seeing this, one can clearly feel that, okay, they have some kind of Iranian ancestry and some kind of Pakistani ancestry. But this is not about the Pakistani ancestry. This is about the South Asian ancestry and how much ancestry is present that comes from the admixture analysis. Here you can see this dark blue color is nowhere present in other Indian population except the Parsi population. And then the Parsi of Pakistan and Parsi of India, they are showing almost the same kind of genetic pattern, even living into two different geographic regions. And also one can clearly calculate this was the South Indian related ancestry, this was Iranian related ancestry, and one can clearly see that there is a very high amount of Iranian related ancestry was present among the Parsi population. So Parsis were one of the group who came nearly 1200 years back from the Iran, and still they have kept that Iranian ancestry even by mixing culturally with the local Indian population. And this was the most interesting observation for the Parsi paper, that the Parsis of the present, the Parsi of present gene pool living in India, they are more closer to ancient gene pool of Iran. They were not more closer to the present Iranian population. The reason is because Iranian population, they got admixed with the other population, probably associated with the Islamic conquest. Otherwise, the Iranian population, they share less number of alleles comparing to Iran Neolithic population that was nearly 10,000 years older. So Parsi still carry that ancestry that was present among the Iran Neolithic population. And when you go further, then there are more complexity of the analysis, and this complexity comes from these kind of haplotype analysis, that you make a bin in the genome, break this genome into various parts, and then compare these smaller parts of the genomes with each other, and then they say that how much is the donor and recipients. You compare each population as a donor, each population as a recipient, and then see that who has given to whom. For example, like the Parsi population of India as well as Pakistan, they received more. These are the donors, these are the recipient. They received more genomic segments from the Gujarati and Sindhi, whereas they have given less genomic segments to the Gujarati and Sindhi population. So this is one of the very interesting observation. Here you can see that it means that they have received more gene flow from the local population comparing to giving their genes to the local population. And the inbreeding coefficient, you can see that they are quite inbred, and their population it is also interesting to see that the education in some population, that is the main causal factor for their reduction of the population. That Parsi, they educated so much that they are not interested in making the babies. So this is why the Parsi population has reduced 120,000. They were in 1940s. In 2011, they are 57,000. In 2023, they are nearly 40,000. So this population is reducing, and that is not because of any genetic problem, that is because of the cultural problem. And the time was, the time calculation was matching with the putative migration of the Parsi population. So this is the last example that was related with the Sri Lanka. Everyone knows there is a very serious ethnic conflict in Sri Lankan population. So we have analyzed two groups, that is Sinhalese and Tamils. These two groups, they speak different languages. These two groups has very serious ethnic conflict. They don't intermix at all. But what we see in the principal component analysis, in the admixture analysis, that we can't differentiate based on their genomic ancestry. So these two populations, they are mixed so much genetically that they can't be differentiated by using simple methods, that is principal component analysis and the admixture analysis. But finally, when we started to compare the smaller segments and the larger segments, the IBD analysis, then we see. So also some of the histories when it comes to the reality, then you can check that which history is correct and which history is wrong. For example, there is a migration in reality, this is the true migration. There is a migration from this region to Sri Lanka. So King Vijay has moved nearly 2,500 years back from here to here, and he started living in Sri Lanka. And then there was another population speaking Dravidian language. They also moved at the same time, and they are also living in Sri Lanka. So these two populations, before the ethnic conflict has started, they had mixed so much that their DNA is nearly similar. But the history says that another version of history that was more popular, that says that King Vijay came from West Bengal to Sri Lanka. And then because of this version was most popular, we never read about this version. So we were comparing the population living in Sri Lanka with the West Bengal as a putative ancestral population. And then we tried, we could not find anything excessive with this population. Finally, we tested another version where we could see that the Sinhalese, the population who has migrated from here, they had just 16% more higher DNA segments comparing to the Tamilian population that was connecting them with the Maharashtra population. So multiple version of histories can be also compared, can be also checked with the genetic analysis, and that is the power of the multiple number of analysis with the genes. So this is my group working in the BHU. So not only my group, there are multiple groups, basically my ancestral, not ancestral, I have done my PhD only there. So Institute of Genomics in University of Tartu. Then from University of Colombo, people were involved, University of Calcutta. Peter Underhill from Stanford University has helped us to sequence a lot of Y chromosome. Then we have a linguistic expert, Professor George Von Dream, as well as our group from CSIR CCMB, where Neeraj Rai is in BSIP now. So we received some fundings from multiple agencies, but we are still waiting more fundings from the Indian agencies. So thank you very much for your kind attention. Parsi? Yes, they do.
But you said the reduction in the population is majorly because of their culture. So inbreeding is not responsible at all? Or?
No, you see that there is not just a single factor. There are multiple factors. There could have been, not much has been studied on their Y chromosome, that if there is some problem with the infertility as well. But so far the detailed information which comes from multiple studies that clearly says that the culture is number one. Second could be their inbreeding, but inbreeding is also high in many population of India. Even you can compare with the Andaman Islands. There are 98 Onge people, and they are increasing step by step, slowly. Even during the tsunami, people have thought during 2005 that all the Onges, they have died. But after a few days back, when water went down, they came back again. So, and these people, they had the prior information that something is tsunami kind of thing is coming up. So they went at the top hill of the Andaman. So, so the population usually, naturally, these population are surviving since 50-60,000 years back. So the reduction, the major reduction for Parsi is not because of some biological reason, that is more with the cultural. Yeah. Or? So for multiple studies, we have used multiple sample sizes.
I saw some data of caste.
Yeah.
Different castes.
You see, that depends on the question. For example, like if someone wants to do analysis of a caste which is practicing a high level of endogamy, then even your sample is enough to tell the story of that whole caste, if you are belonging to the same caste. For Parsi, there were multiple samples, nearly hundreds of the sample we have used for Parsis. And that was for the genome wide, for 300 sample has been done for the mitochondrial DNA. So, different number of samples for different kind of analysis. The sample calculation. Usually, we don't use any statistical method for sample calculation because for the autosomes, one can see that if that particular population is practicing the endogamy, then one sample is enough. That is practically one sample is enough to tell the whole story of that population. But nearly for genome-wide analysis, we use 20 samples for the whole population. And for mitochondria and Y, we use more than hundreds of the sample for mitochondria and Y. There is no hard and fast rule to use number of samples for ancestry information. For example, like for ancient DNA, the Indus Valley samples, this was just a single sample. But that has given much more information that we ever have based on the modern DNA samples. Yeah. Sorry, one more time, please. Yeah. Okay, just repeat. Yeah. Sinhalese and Tamil. Yeah. No, we used all the PCs, and we could not able to resolve this similarity. We used the IBD, and this IBD was able to tell only when we took some ancestral population that was a Maratha population from Maharashtra, that 16% more number of smaller IBD segments were present among the Sinhalese comparing to the Tamils. Yeah. Otherwise, they are quite similar. This was also, a nightmare for us. We collected multiple number of samples, assuming that maybe same population could have been sampled multiple times, but result was the same. Reading about the ethnic conflict, but that has started just few hundred years back. Previously, the population was mixing very vigorously. Yeah. And there is another third population, Veda. And that population shares more ancestry with mainland Indian population comparing to the Sri Lankan population. So that is another complexity with the South Asian population. At some population, they share more. Another population living in the same vicinity, eating the same food, but do something, their ancestry shows something drastically different. Yeah. Thank you.